at the University of Connecticut Health Center. And Alexandra's going to talk about GABA2 genetic variants and chromatin accessibility in induced pluripotent stem cell-derived neural cells and postmortem brain samples in the context of alcohol use disorder. Okay.
Okay. So thank you for already saying the very wordy title, so I don't have to say it. I'm currently an 8th year MD PhD candidate at UConn Health in the laboratory of Dr. Jonathan Kowal, and I'm part of the Alcohol Research Center here. And over the last 4 years, I've been studying various genetic endophenotypes that are possibly correlated with increased genetic predisposition to developing alcohol use disorder. And now I can't advance. There we go. Alcohol use disorder is defined by the DSM-5 by a set of 11 criteria, which range from not being able to reduce the frequency or the amount of alcohol consumed, having alcohol-seeking behaviors interfere with various commitments in the community, whether that's to your family, friends, work, etc., putting yourself or others in physical danger due to your alcohol consumption, and experiencing physiological tolerance or withdrawal to the substance. Approximately 8.5% of adults in the United States are afflicted by moderate or severe alcohol use problems, which is meeting 6 or more of those 11 criteria. Excessive alcohol use is the 4th leading contributor to disability-adjusted life years, or in other words, the loss of healthy life years in which one can meaningfully contribute to the community in the United States, and costs the country's economy upwards of $250 billion per year in medical costs, repair costs after automobile accidents and other physical harm, etc. Twin studies suggest a 50 to 60% heritability of alcohol use disorder. And one of the genes or sets of genes involved in genetic predisposition to alcohol use disorder belong in the GABA-A receptor subunit gene family. GABA-A receptors are the primary inhibitory ligand-gated ion channels in the central nervous system, and it's been shown that ethanol potentiates GABA-A mediated signaling. TAG-SNP markers, which essentially mark larger inheritable blocks of the genome, termed haplotype blocks, in the GABA alpha 2 subunit gene, have been associated with alcohol use disorder in multiple case control studies, and more recently in a meta-analysis of over 3,000 individuals. Alpha 2 is one of 4 GABA-A receptor subunits on chromosome 4p12, and it's been shown previously that alpha 2 containing receptors are responsible for mediating the anxiolytic effects of benzodiazepines, as that's where the binding pocket for those drugs is located. In mice as well as humans, GABA-A receptor subunit genes are organized into clusters. And that is shown by my poor artwork on the left. And as I mentioned, the cluster that we're interested in is chromosome 4p12, which contains the genes GABA gamma 1, GABA alpha 2, GABA alpha 4, and GABA beta 1. And the arrows indicate the direction of transcription. More specifically, the alcohol use disorder associated haplotype block is marked in red, and that extends from the middle, approximately, of GABA alpha 2 into the intergenic region between alpha 2 and gamma 1. And down at the bottom is a schematic of a typical heteropentameric GABA-A receptor, which functions to allow chloride to move down its concentration gradient. Separately from being associated with alcohol use disorder, variants within this haplotype block have also been associated with various neurophysiologic endophenotypes, including reward anticipation and insular activation. This suggests that variations in alpha-2 may have subtle effects on neurodevelopment. Importantly, for you to know, there are no coding variants in alpha-2, and thus it's been hard to determine the mechanism by which variants in the AUD-associated hepatotype block affect gene expression. The public health significance and basic science significance of this project is identification and characterization of functional variants within GABA alpha-2, and more specifically within the AUD-associated haplotype block, could aid in better understanding of neurodevelopmental pathways that increase vulnerability for alcohol use disorder. And this could subsequently aid in counseling of people that are at increased genetic predisposition for developing this disorder, and could also possibly aid in forming more specific treatments for this disorder. The model system that we use is induced pluripotent stem cell derived neural cultures. So how we generate these is people who have participated in clinical studies studying alcohol use disorder have generously donated dermal fibroblasts. These are sent to the stem cell core at UConn and reprogrammed using the Yamanaka factors to produce iPS cells. And over the course of 12 weeks, via an embryoid body differentiation protocol, we generate neural cultures which express neural specific genes and are electrophysiologically similar to neurons that you would isolate from a brain. Previously, we've reported that there is a correlation between the genotype at a tag SNP RS279858, which lies in exon 5 of alpha-2, and the expression level of the GABA A receptor subunit genes on chromosome 4p12, with the increasing number of C alleles being associated with a lower level of expression of these genes in the cluster. In a manuscript that was accepted for publication last week at the American Journal of Medical Genetics Part B, we added to this picture by studying the promoter proximal methylation at the 4 genes within the chromosome 4p12 cluster, and found that there was a significant and additive effect of methylation status at GABA alpha-2 and genotype at RS279858, again within the gene GABA alpha-2, such that the more C alleles a sample had and the higher the methylation in a given sample, the lower level of beta-1 expression in neural cultures. And this was only significant for beta-1. What's fascinating is that if we look at this linear schematic of the cluster down at the bottom of the slide, alpha-2 and beta-1 are approximately 600 kilobases apart. So we wouldn't expect there to be any sort of correlation if they possibly didn't physically interact in some way. This idea is supported by virtual circular chromatin conformation data, which was collected by Schmidt et al. And put into this repository at the Way Lab that's publicly available. And when we look at specifically at the brain, this only happens in the brain and not in other tissues. When we choose our anchor point as RS279858 to represent the AUD-associated haplotype block, we see a peak. We see a peak at beta-1, suggesting that there is a brain-specific interaction between the AUD-associated haplotype block and beta-1. This allowed us to form a model, which I showed 2 years ago in a talk that I was very grateful to give to the society, such that there are allele-dependent long-range chromatin interactions that are brain-specific that could underlie the association of beta-1 expression with the tag SNP RS279858 and ALF2. And again, I already showed the linear schematic, and we're suggesting that there may be an increased interaction with the AUD-associated haplotype block in the case of the T allele, resulting in higher expression of the chromosome 4p12 GABA A receptor subunit genes, and a lower level of expression due to reduced physical interaction between genes within this cluster and the AUD-associated haplotype block. To add to this picture, I've performed some ATAC-seq, or the assay for transposase successful chromatin. And here I'm only showing two samples, one which is a homozygous CC sample, and one which is a homozygote TT sample, and we see a peak present in the CC sample and not in the TT sample. And these are both iPSC-derived monolayer neural cultures. And down at the bottom are the raw reads. When we combine the data on the previous slide with information from the genome browser, we find that this region overlaps a digital genomic footprint in fetal brain that's pretty strong, as well as a brain-specific enhancer-like region predicted by chromatin HMM, which is a composite of multiple histone marks, and notably is also in proximity to a number of other single nucleotide polymorphisms that are in linkage disequilibrium with RS279858. We've taken this observation and talked to the genome editing core, and they've generated a 1 kilobase deletion that encompasses this digital genomic footprint, the enhancer-like region, and the SNPs that are in LD with RS279858. And I'm in the process of differentiating those into neurons to subsequently assay via ATAC-seq to see if deleting this region alters chromatin accessibility, and subsequently we'll measure gene expression to see if the loss of this region alters gene expression of the genes in the chromosome 4p12 cluster. In conclusion, methylation analysis of alpha-2 and beta-1 suggests an additive effect of methylation at the alpha-2 promoter proximal CPG island and RS279858 genotype on beta-1 expression in neural cultures. And this, as well as the publicly available 4C data from the dorsolateral prefrontal cortex, as well as other postmortem brain samples, suggests the presence of a neural-specific long-range interaction between alpha-2 and beta-1. Preliminary ATAC-seq results suggest the presence of allele-specific regions of open chromatin, one of which I showed you today, and the other that is mentioned in the abstract. And differential accessibility to various transcription factors could be partially responsible for the transcriptional endophenotype that we observe that's associated with the RS279858 genotype. And as I said, we're performing ATAC-seq in the edited lines, targeting 2 different regions, one of which I showed you, and the other, again, which is mentioned in the abstract, and I cut out for time reasons, that are both in LD with RS279858 to assess whether editing these regions affects chromatin accessibility in neural cultures and subsequently affects gene expression of the chromosome 4p12 genes. And ultimately, we would like to perform 4C-seq on our own samples to confirm the presence of a physical interaction between alpha-2 and beta-1 that's neural specific in nature. Finally, I'd like to thank the people that were involved in this project, especially my advisor, the people who have been involved in the past in generating cell lines and such. My committee, Center for Genome Innovation, who's been responsible for sequencing a lot of the ATAC-seq processed samples, the Genome Editing Core, and especially the financial support to both my advisor, my pre-doctoral fellowship from the National Institute on Alcohol Abuse and Alcoholism, and the program grant to the UConn Alcohol Research Center.
All right, thank you so much, Allie. Unfortunately, we don't have time for questions now, although I would be interested in picking your brain about the functional implications of that beta 1 and alpha 2 interaction. So maybe another time. Now we're going to move on to our next speaker, who I'm excited to introduce because she is currently a graduate student at University of Illinois at Chicago in the lab of Amy Lassick, where I did my graduate work. So Kana Hamada will now be talking about the effects of ALK and STAT3 signaling after binge drinking. Are you ready, Kana?
I am. Thank you, Antonia. As Antonia mentioned, I'm in the Lassick lab at UIC, and today I will be talking about the activation of ALK and STAT3 signaling in the mouse prefrontal cortex and hippocampus after binge drinking. So very briefly, alcohol use disorder is a chronic relapsing psychiatric disorder that affects 15.1 million individuals in the United States. It is a serious health issue with high personal and societal costs. The estimated financial burden associated to AUD in 2010 was $249 billion, of which 3/4 was attributed to binge drinking, which highlights how detrimental binge drinking can be. Binge drinking, as defined by the NIAAA, is a pattern of drinking that brings the blood alcohol concentration levels to or above 0.08 grams per deciliter or 80 milligram percent. This typically occurs after 4 drinks for women and 5 drinks for men in a 2-hour period. We model binge drinking in mice using a well-known drinking in the dark paradigm, where after 2 weeks of acclimation, mice are given access to 20% ethanol 3 hours into their dark cycle when they are most active. And through this paradigm, the mice voluntarily consume physiologically relevant levels of alcohol. As you can see in this third graph here, both male and female mice consume enough alcohol to bring their blood ethanol concentrations to or over 80 milligram percent. One of the targets that we have been studying in the Lassick lab to understand drinking is anaplastic lymphoma kinase, or ALK. ALK is a receptor tyrosine kinase that is expressed throughout the brain. Variants in the human ALK gene are associated with changes in alcohol response, and our lab has shown that both systemic and localized inhibition of ALK reduces binge-like drinking. One of the targets of ALK that we are focused on right now is STAT3, or a signal transducer and activator of transcription 3. STAT3 is a transcription factor involved in immune signaling. Our lab and others have shown that STAT3 is activated by ethanol, and in vitro, our lab has shown that STAT3 is activated by ethanol in an ALK-dependent manner. Which leads us to our hypothesis. We know that alcohol exposure activates ALK, and ALK inhibition decreases binge-like drinking. So we hypothesize that STAT3 acts downstream of ALK upon alcohol exposure in vivo, and that the inhibition of STAT3 will decrease binge-like drinking. One of the first things we did to understand the involvement of the ALK-STAT3 pathway was to look at ALK activation as measured by tyrosine phosphorylation immediately after the final drinking session. By doing so, we observed a significant increase of phosphorylated ALK, both in the prefrontal cortex and in the ventral hippocampus. We next thought to look at STAT3 activation again by looking at tyrosine phosphorylation, and unfortunately did not see any statistically significant changes. And this could suggest that ALK is not activated, or potentially that STAT3 activation is more transient and we missed the time point, or we missed the time of its activation. STAT3 could be activated in a subpopulation of cells, and then this result and that change was just not observed in the way that we collected our tissue. But while we didn't capture STAT3 activation, we do see a very strong and positive correlation between phospho-STAT3 and phospho-ALK expression only in animals that consume ethanol in both the prefrontal cortex and hippocampus, which suggests to us that ALK and STAT3 may be co-regulated by ethanol. And so then we took another approach to study the involvement of STAT3 in binge drinking by looking at the expression levels of some of STAT3's well-known targets, such as GFAP and SOX3. We found an increase in these STAT3 target genes, SOX3 in both the hippocampus and prefrontal cortex. We found other increases in STAT3 target genes such as LCN2, TNF receptor alpha, and GFAP, which led us to our next question, which was, what would happen if we manipulated STAT3 activity and whether that would have an effect on drinking behavior. To test this, we used a STAT3 inhibitor called Static, which we administered at a dosage of 20 milligrams per kilogram subcutaneously half an hour prior to the second, third, and fourth drinking day, here denoted by the blue arrows. We found that systemic STAT3 inhibition significantly decreases binge-like drinking in both our male and female mice, as seen by a decrease in these black dots in 2-hour drinking, in the 4-hour drinking, and in their blood ethanol concentration. The same dosage of Static does not significantly decrease sucrose or water consumption. Finally, to understand the role of the ALK-STAT3 pathway in alcohol use, we use the NIH Links 1000 database to mine for novel genes that may be involved. This analysis was done in collaboration with Dr. Laura Ferguson and Dr. Dane Mayfield at UT Austin. So first, what is the Links 1000 database? The Links 1000 database is a project that characterizes perturbations in cellular signatures after small molecule application. So the L1000 project collects gene expression profiles of 1,000 genes for hundreds and thousands of small molecules on cell lines at different doses and at different time points. For our analysis, we mined the database to compare the transcriptomic signatures between ALK inhibition and STAT3 inhibition with the transcriptomic signature of ethanol consumption to generate a list of genes that are both regulated by the ALK and STAT3 pathway and altered by ethanol. This is the schematic for the bioinformatics approach that we took. So first, we looked at the database and found 2 ALK inhibitors, NBP-TAE684 and alectinib. We took the overlapping transcriptomic signatures between our 2 ALK inhibitors to generate our master ALK inhibitor signature. After that, we compared the ALK inhibitor signature to STAT3 inhibitor niclosamide's transcriptomic signature and took this overlapping gene set to be our ALK-STAT3 inhibitor signature to denote a group of genes that are regulated by the ALK and STAT3 pathway. Finally, we took this ALK-STAT3 signature and compared it to the gene expression changes that were observed in the prefrontal cortex of female mice after a prolonged GID session. This overlap then was our gene set of 95 unique genes that are potentially regulated by the ALK-STAT3 pathway and also altered by ethanol consumption. To investigate the genes from our gene set, which genes may be directly regulated by STAT3, we performed a STAT3 binding site analysis in collaboration with Dr. Mark Meschenklein at UIC. And the following were our analysis parameters. We took 2 kb upstream of the transcription start site to be our promoter region. We found the consensus STAT3 binding motif through Jasper and used the FindMo analysis tool to search for STAT response elements in this 2 kb promoter region. By doing this, we found that 53 of our 95 genes had putative STAT3 response elements, totaling a number 91 total motifs identified, which is a very high percentage of genes with putative STAT response elements. The genes on the slide here are just some of the examples of the genes that have STAT response elements and their locations in the promoter region. So finally, we were further interested to see if these 95 genes that we found through our bioinformatics approach were involved in any known networks. And to understand that, we used the Ingenuity pathway analysis in collaboration with Dr. Harish Krishnan, again at UIC, and the network analysis yielded 7 networks. Here I am showing two of our top networks. The first network denoting a cluster of genes that are involved in cellular compromise and tissue disease. The second network are a cluster of genes involved in cellular assembly and organization. From these top two networks, we took several genes to validate by qPCR, here denoted in green. I'm not sure if you can see them. And we validated in the brain regions, the hippocampus and prefrontal cortex after just 1 round of binge drinking, and found increased expression of these novel putative STAT3 target genes after binge drinking. We found an increase of NR1H2, SMARCA4, SMARCC1, and GPNMB. To summarize, I hope we've shown that binge-like drinking activates the ALK and STAT3 pathway. Systemic inhibition of the STAT3 significantly decreases binge-like drinking without altering sucrose or water consumption. We have potentially identified novel target genes that are downstream of the ALK and STAT3 pathway that are upregulated by ethanol consumption. And with that, I'd like to thank Amy, my lab members, our collaborators, of which we have many, my thesis committee, and our funding resources. Thank you. Okay.
All right. Thank you so much, Kana. We do have a question from Carla Khan. Okay. And that is, do you know if other stats are similarly affected and or if manipulating STAT3 affects the other stats and their binding targets?
Ooh. We do not know, and we actually haven't looked at the other stats. It's a very interesting question. Yeah.
I have another question. I think we have just that time for one question. I know that the LINCS database uses several different cell types, correct?
Yes.
But you were able to replicate this in the hippocampus. I'm wondering if the effects on these downstream target genes you would expect to be sort of universal effects, or if you think they are specific to the hippocampus and PFC.
So like you said, the LINCS database uses primarily cancer cell lines, and there are not very many like neuronal or any like glial, like just any like gliogenic cell lines either. And so it's pretty hard to say, since we didn't look at any other brain regions, if this effect is particularly universal. Like, I don't think there are regional expression differences in STAT3. So it would be very hard for me to be accurate, like confidently give you an answer to that.